dissection and identified loci that control seed protein content and water use efficiency. He is also on the team of scientists that sequenced the soybean genome, and he is a fellow of the AAAS, the CSSA, and the ASA. So with that being said, let's give a warm welcome to Dr. Specht.
Can everybody hear me out there? So, okay, thank you. It's a pleasure to be here, and I do want to thank all the sponsors. They're listed on the back of your program here, and especially the graduate students who invited me to speak. So every once in a while during my talk, you may see the inflection in my voice raise a little bit because I do that, and I did that in class. That was to keep the students awake, so I hope you don't mind that. It's not really the reason I did it. It's to emphasize the point. But I think what I'm going to do here is add a little bit of humor in places, because some of the things I'm saying you're going to be happy about, and other things you're going to be glum about. So let's get started. First of all, this presentation is really on behalf of the hundreds of soybean breeders in the world and the USA, because what I'm addressing are their accomplishments. Just a little bit about funding. I don't have USDA on here, and they gave us a fund a long time ago approved a grant a long time ago that got our marker work with Perry Cregan and Randy Shoemaker, and I started, but since then we've been supported by USB, Nebraska Soybean Board, North Central, and of course the University of Nebraska. Now, soybean is a relatively young crop in the U.S. You can see this is the harvested acreage on this side, acreage on the outside, hectares on the outside. And by the way, I'll have both metric and English units on most of my slides. But you can see they really didn't start taking off in the U.S. until the late 1930s and 40s. And then they really took off until they hit a peak in 1979, and then thereafter they fell a bit, and now they're back up to where they were. But I put these red arrows over here to indicate that Brazil is now beating us in those situations, okay? This is what, if you remember, if I go back, this is an arrow here, and I'll show you the two maps. This is what the production was, harvested acres, generally most in the U.S., 1979, 80, that's what, 23 years ago. That's what it was. You can see there's a lot of packing of acreage down here and really little there. But now you see the change. In fact, because of global warming, I don't show you a Canadian map here, but they're growing almost as many soybeans now as we are in the northern tiers. Now let's just get right to the point. Here's the trend line from 1924. This is on-farm soybean yields. This would be recorded by the NAS people down here. And this, obviously, each of these points is kind of like a normal curve of farmers that report in. So you'll have some that have greater yields than that and some that. But this is the mean for the U.S. averaged over all the states. That year is down here, seed yield, bushel per acre, English metric over here. And I've got both equations in English and metric here. So if you look at this particular trend line, it's 0.346 bushels per acre per year. That's the total for all on-farm improvement, including the breeding inputs and the agronomic inputs, plus the interaction between them. So when I first did this, so this is what this average, I put this number up here as a final, that's what this line represents. So when I started looking at this area back in 1981, I went back and got my data and replotted. I want you to watch this number because this is the linear value on the seed per bushel axis. Of course, this is the linear there. But this was the report I made in 1981. And just watch this line and look at this number as I go through it. So I made this report at the SA meetings in Atlanta, Georgia. Note that number jumped up at 1998 when I reported the same numbers with more time in Baltimore. And when I presented this a couple of years ago at the STA meetings in Chicago, you see it jumped up here. So I'm going to back up just again so you can see that. Watch this number right here, 31, 33, 35. That to me tells me there's a little bit of curvature in that line because it's increasing over time. So the one thing we have to keep reminded of is, this may be the glum part I was going to talk about, there is a biological yield limit established by first law of thermodynamics when you're converting light energy into food. Now the question is the efficiency with which you can do that. That's another story. So really what we should be looking at is actually logistic projections, and I'll give you just a common example down here where the key features are, okay, what's the maximum you can get with the inputs, and then of course the inflection point where we go from acceleration to deceleration, and of course where that occurs left to right, okay? And the K value, of course, is a constant, or not the K value, but the R is a constant. So let's go back. Here again is our linear line. If you do the math, you'll see it up here in ProcSAS or anything, you can use the information criteria, which assesses the goodness of fit of a model relative to the complexity of that model. You want to explain as much as you can, but also use the minimum amount of parameters. This is a law of parsimony, Occam's razor, so to speak, okay? So let's take a look at that. When you take that data set, and that data set only, no other assumptions, and assume a logistic projection. So this is what you come up. So you're using the same data, but you have a logistic model here that I just described, and as you can see up here. And the AIC criterion here says that this model is a better fit than the linear model. And this is a famous equation down here from math in terms of by the Japanese guy that's listed down there, the name of this. So I'll come back to that later. So you can see this number up here is 141. If you decrease that number, this model is better because it takes into account both the model complexity and the amount of fit. Now what I've done also is try out another model. So basically what we're looking here is this part of the curve. And what that curve says that the inflection point here, based on a simple model, is going to be in 2008. That's past us already. So we're 5 years or more onto this side already. But we can't, the model doesn't predict that, it only predicts this first part. So, and I also noted that there's more environmental variability here with climate change or whatever you want to assume. And that if you use a segmental model, which is only the first part of the logistic curve, but with a breakpoint, you have a yield before the breakpoint and a yield after, and then you determine what year that breakpoint is. And so you see this number, the ACE criteria number, is actually lower. So it's the best model given you have here. So apparently in the course of on-farm soybean improvement, we were steady here, and then because of some technological advancement, either genetic, agronomy, or both, we've jumped to a new level. So you can see right here, it was 0.32 bushels per acre per year here, but now it's 0.438. Okay. So obviously we've had some technological input that has improved yield. But let's just take a look at where that comes from. One is genetic technology, soybean breeding, if you will, continual release and rapid adoption of producers, more yield potential, protection of current yield, as the prior speaker talked about. Agronomic technology, obviously that's improved yield too because you have more effective crop management system, decision aid tools, and field equipment. And I think what we all forget, either breeders or agronomists, is there's synergism here that applies. Whenever you have an interaction, it's not necessarily negative if the fact that the modern agronomy and the modern genetics connect with each other to give you more yield than you would expect based on additivity alone. So what a breeder's goals are, that was mentioned by the prior. I list them as productivity. I want more yield potential per se. And I distinguish between yield potential and yield here because you want to develop varieties that will yield better in any environment, but their potential may be suppressed in some. Okay. Likewise, protection down here, and quality. This is something Tom Clemente does well here. He's actually making soybeans ready for fish food, right, Tom? Actually, he wants to feed all breeders to the fishes, so just a joke there and there. But the other thing that you notice, I don't list abiotic stress up here because any factor I figure that's a resource for crop consumption really can't be called a stress. You just don't have enough of it. It's not a stress, in my opinion. What other factors? Rise in CO2. It went from 315 when I was in junior high all the way to 396 and actually touched 400 here, and it's November high and May minimum. And I've calculated, and others have too, that it accounts for about 0.05 to 0.07 bushels per acre per year. Not very much, but constant. Multiply that by 40 years. Bingo, you got a pretty good number. Also, soybean, and this doesn't apply to other crops, soy-corn rotation is replacing continuous corn, and actually both corn and soybean yields are better off for it. Because they'll go up, and of course soybean gets on the better ground that corn used to dominate. And of course this is all the critical one down here that we refer to. That's we need funding to do both curiosity-driven basic research and mission-based applied research, because those discoveries are eventually translated into R&D that benefit the consumers, who are the producers. So let's take a look at this. How much genetic technology underpins on-farm? So you need a knowledge of what the annual rate of genetic gain is and compare that with the on-farm gain. And so we did an estimate of that recently. I'm going to step back a little bit to show what we knew in the past. Did a study back in '84 where we looked at about 200 cultivars, and we measured their yield. This is the release year down for the release year of the cultivar. This is the yield of the individual cultivars by dots. And these, before 1943, were basically plant introductions brought from China, improved, purified, and then released to farmers. So they're just Chinese lines that were brought here and used. After that, soybean breeders decided to adopt what corn breeders were already doing for a while, and that's make matings and try to generate crosses or progeny from those matings that were superior to both parents. And so in effect, you see, if you look at these regression lines here and here, here and here, what you see is basically a quantum jump occurred when they did that, when they shifted from plant introduction to plant breeding, basically. Equivalent to the switch from open pollinated corn to hybrid corn, in my opinion. Okay. Here's just the bottom line value. I said it amounted to 25% average over all these maturity groups. Not a bad figure for a quantum jump. Wilcox was the last one to publish something on this before we did, and he gave us this values here. And again, on-farm improvement is 0.35 bushels per acre, and this is the conversion factor. So those numbers up here that he's given translate between 0.32 and 0.46 if you do it on a bushel per acre basis. So they're in line with the on-farm improvement, but I'll come back to that. Soybeans, we did an experiment. Soybeans, of course, are maturity group dependent. They have to be adapted because of the photoperiod and flowering. Corn, you do it on a GDD basis, but in soybeans, we do it on a maturity group basis. You can see there's a series of maturity groups here, depending on the latitude and what photoperiod we have in spring and midsummer. We're going to concentrate only on these three for the study I'm going to describe next. That's the 2011 data. And these areas right here probably account for the majority of soybean production, but these account for a significant other. Right now, those others in terms of yield at least, so the total three account for about 75 to 80% of soybean production. So what we did is we took maturity group 2, 3, and 4 soybean cultivars that had been released back in the 1920s all the way to today, and they were both public ones and proprietary ones contributed by Syngenta, Monsanto, and Pioneer. Now, because of an MTA, I'm not going to tell you their names, but you're just going to see black spots for everything, okay? Dots. So the maturity group 2s were grown at these locations, one of which is in Nebraska at Mead, and of course, and some in Canada over there were 2 adopted. There were another set, whoops, went the wrong way here. 59 or so, there were 60 before, 59 grown here in that zone, at those locations, 15, and then maturity group 4, there was another down here in those regions. So we were growing these 2s, 3s, and 4s, these historic sets, at the locations they were adapted to. Here's the genetic gain. A lot of soybean breeders and agronomists here. We also took the opportunity to use that information. Brian, I, and Dave Sleeper got the test organized, and Randy contributed the old varieties, and Keith was a graduate student that worked for Brian Deers that helped us summarize it, and George, of course, was in this project too. So what did we come up with? Again, a plot of seed yield versus release year. Those are the circles, are the maturity group 2 averages over all the locations they were tested in 2 years. And so what you see down here, this is the straight line. If you use a linear regression, the broken line is the segmental linear two-segment model. Okay. So you can see that. You can see if we use the breakpoint model, it shows that the breakpoint occurred about 1968. But those have standard errors around them, okay? You also can see that if you apply the AIC criteria, the segmental model is 99% better according to the AIC criterion than the linear model. So this is what we have to assume. Here's maturity group 3 data. The breakpoint here was 1964 plus or minus an error around that. And again, you can see that the breakpoint here model is much better. It's 97% better than the other when you do the AIC criterion. Here's maturity group 4. You didn't see how it fell down here, but I'll show you that in a minute. It also shows a break around 1971. And again, not as much as support for the segmental model, but clearly about 5 times greater support. So that's the data. If we put them all together, you see this, and you see this as the breakpoints. Anybody want to yell out what the year the Plant Variety Protection Act was passed? 1970, right? Okay. Pretty good fit here with the data because we're checking the original varieties. The breakpoint for the U.S. was 1983, but you have to allow time for these cultivars to get out there before their impact shows up. So it might be 10 years later in the on-farm yield production before you see it. So clearly you see something here. You see maturity group 2 and 3 are pretty good, but 4 is a little lower, and 4 is down here. And of course, the yields are highest if you look at these numbers down here, right, in 2s and 3s. The maturity group 4 zone right here is actually not that good a place to grow soybeans. And here's the data that shows that if I break out the U.S. data by states, you can see we're back to the 0.3461 bushels per acre here. Just considering the maturity group 2, 3s, and 4s, they pretty much, because they occupy 80% of the acres, they're pretty close to the USA. But if I break out 2s and 3s from 4s, you can see that 2s and 3s are better than the U.S. And that's why soybeans migrate. They migrate to the area where they're most profitable and most yield, right? Except for other considerations of the farm program. Now, all breeding has consequences, no matter what you're doing. So the improved yield by 1/3 of a bushel per acre per year, what you see is the protein goes down, the oil goes up. Now, as much as people like to say my variety yields has protein as good as the rest, they're not dealing with long-term data, so they can't show that. Of course, I don't show all the dots here, but the question is, are those outliers and don't repeat the next year? So the key question here is that protein has gone down. There's a reason for that. Protein is more expensive for a plant to make than lipid. And the physiologists that published a few years ago that said lipid should be more expensive than protein were wrong. They didn't figure out that storing protein in a seed requires a lot extra energy beyond just equations compared to storing lipids in lipid lined membrane bodies. So it's not surprising then that protein would go down and yield goes up when you're putting selection on, or oil goes up when you're putting selection on yield. So Brian looked at these data, my cohort who were publishing this, he said, okay, we have 23 kilograms per hectare per year yield here, and this is what breeders have done. Breeders have underpinned all on-farm improvement. Now, soybean guys out there, don't break your arm by patting yourself on your back, because I'm going to tell you that ain't true. Okay. So let's move on to that. So this is the genetic gain rates, and that's another issue about genetic gain. It's entirely dependent upon the environment in which you measure it. So these are the low environment estimates of genetic gain, and these are the higher environment estimates of genetic gain. What you see, as you allow for more expression of genetic potential in these better environments, you're seeing that your rates go up, which implicitly tells me we have much more genetic potential in these varieties than we do down here. Well, I shouldn't say these varieties, because these are the same varieties that were used to get the genetic gain. All of them used to get the genetic gain in maturity group 3, 4 here. All those 49 and so on as you go. So each point represents, within its maturity group, all of the varieties and the computation of the genetic rate of gain for those varieties, say at this very low level here in our test environments across the USA. So hopefully I made that clear. So this is a single location estimate of genetic gain for all of the maturity group 4s, just as this is the same varieties tested for genetic gain for all maturity group 4s. Okay. So you can see the genetic rate of gain that you come up with is entirely dependent on the potential that's allowable to be expressed in an environment. Now, breeders have long been in a love affair with this, and I don't know the reason why. I do know the reason why, and I thought, well, now we'll have a chance to prove why it's useless, okay? So Brian put these slides together, and he indicated in most cases, if you measure a single cultivar out of 20 or 30 or 40, and then you use the rest of the cultivar means to estimate their yield, so you can plot that cultivar's yield against all other cultivars, you get these kind of regression coefficients. And the thought was that a stable one was more useful as compared to one that was less stable or less stable on both sides. Okay. So I said, let's test that. If we look at the stability coefficient for each variety, well, each of the releases over the years, did stability change at all? His hypothesis. Mine was new cultivars are actually less stable, but that means they're more productive. Okay. Because obviously the coefficient there is changing, going from wherever 1 is on this scale to greater than 1 or less than 1. Okay, because we're calculating the stability coefficient, not the yield. And so the question is this the kind of stability analysis you get where you have one like this and one like this? And is it new cultivars versus old cultivars? Or like I was saying to Brian, it's more like this, that the new cultivars beat the hell out of the old ones all the time, except way down here where it doesn't matter, right? You're going to go broke down here no matter what you plant. And so what does the data tell us? So this is the newest cultivars. This is the oldest. You can see right here they don't overlap. This is steeper regression line than this. So this one's going to have a greater stability number than this. Here's for maturity group 3. By the way, each 6 points in there are the 6 cultivars that we determined that overall had the highest yields in the overall analysis versus those that had the lowest. So what you can see here, same thing applies there. And maturity group 4, which is consistent where they're growing the soybeans down in environments that are very low productivity for expression. You can see maybe you'll get a crossover down there. But for the most part, the newest cultivars will beat the oldest ones. And if you go back and read the literature, Rouse had a very good paper in the Journal of Experimental Botany that showed that same thing applied in wheat. So this is what new cultivars, yeah, they're less stable, but I'd rather grow one of these than these anytime. And so would a producer. So here's the stability analysis calculated for each of the cultivars. And you can see as we've moved up in release, the stability parameter, the coefficient, has changed from less than 1 to greater than 1. So that particular Finlay Wilkinson can go in the trash can of ancient history, okay, because what's important is this. All right? And here's again repeated here just so you can see it in color. So I'd like to come back to this. The genetic gain are greater when we measure genetic yield gain in this environment, we get this. When we measure it, we get those, and so on. So as we move agronomics, and maybe my colleague Patricio is here, as we move to higher levels of yield by better agronomic management, we get better expression of the resident genetic yield potential. So I won't go over this compared to old cultivars, new cultivars, greater yield, a little later maturity because we're getting longer summers because of global warming. So our newest releases tend to take advantage of that. And some other things, you've seen some of these. Greater disease resistance, I didn't talk about that, but there's another paper coming out that isolated the ones that were resistant to certain diseases versus non-resistant. Greater total biomass. And the first example of synergism between breeding and agronomics right down there when it comes to planting date. Am I doing time-wise? Okay. And the battery may have gone dead here. I don't know. Not popping up either way. Can you change the page for me? Okay, thanks. So what about agronomic technology? I've already said that you'd be a little naive to assume that all on-farm improvement comes from breeding. It doesn't. So, but this is more difficult to unmingle the changes that have occurred in agronomic improvement with the changes that have occurred in breeding. But so that's working now. So I don't know why it wasn't stuck. I'm just going to talk about one of them. This is a list of it, and you can go to the website to get this listed. But early planting is the lowest hanging fruit on this agronomic practice. So if you look here, study we did, picture date 6/26/2003, 6/24/2004. You know the longest day or the longest day of the year is around June 21st, June 22nd. So the pictures just a few days after that. These are the planting dates of these demo strips we put out there. Early May, mid-May, late May, and mid-June, about 16 days apart. And that's what they look like when we took the pictures. Basically, that year in 2003, if you looked at this sweep across here, these yielded a 1/4 bushel per acre for every decrease in 1 day of planting date. In 2004, which is a better year for growing soybeans, the cost for delaying soybean planting date was 5/8 of a bushel. So if you translate this number into $12 per bushel right now, for every day you don't have soybeans in the ground, you better take $3 out of your pocket and burn it, because that's the opportunity cost you lost by not adopting that. Everybody awake now? Okay. So that's the opportunity cost. Down here it's even greater. That's about $7 or $8 down here. And that's just for a single acre. 1 acre. If you have 1,000, you better take $3,000 out of your pocket and burn it. Okay? And why is that? If you go back here, I'm going to treat these numbers in the colors here, and you're going to see this. This is the photo period during the year from January to the longest day here. In Lincoln, it's 15 hours and 10 minutes. These are the planting dates. And so you can see if you plant here and your crop comes up, your plants are starting to gather light even before the longest day of the year. And remember, the job as a producer is to harvest the sun, because that's the incoming input energy for photosynthesis. You plant here, you're only going to capture what's left of the season. So if you look here, these guys are almost collecting all of the light by the longest day of the year, where these guys over here have hardly started. In fact, if you look here, they're not even covering the rows, so the transpiration and the evaporation will be even higher. So this is when you plant late, that's how much you can use for photosynthesis. So when you plant early, this is how much. That should be pretty clear, I would think. And of course, if you plant later, you're not going to use as much water. But that means you're going to not do as much transpiration. And that means because transpiration and yield are entirely linearly linked to each other, you're not going to get as much yield either. So it's better to use more water if it's available. In fact, if you go back here, you can see once you cover the ground, you're going to capture the rain. Plus, you're going to use more of the rain that comes in earlier that could have run off. And from my colleague Purcell in Arkansas, you can see that when you look at the data here, and we just look at Illinois, it's much more water use efficient to use water with earlier planting. So clearly, the advantage of planting early comes well with the water conditions conservation as well. So crop ET is just defined as evaporation of water from soil and wet surface, but transpiration is a term for the escape of water from soybean leaves. For every CO2 that comes in a stomated soybean in mid-season, 400 water molecules go out. The plants use a lot of water. And so you want to use it as conservatively as possible, but covering that soil surface area to eliminate this, by doing this, to get mostly T. Because T relates to yield, E does not. All right, and farmers adopted this. If you look back in 1980 and you look at the curve of the planting date progress from the day of the season in 1980, They were planting a heck of a lot earlier. In 1912, they were planting a lot earlier. Those are the curves. You go and plot these data points right here at 50% progress, and you get this. So on average, and by the way, Patricia was the one that gathered some of this data for me. What you can see here is producers have advanced planting date by a half a month. So they're not stupid. They know that. But when I look at Illinois data, they're not so much doing that because they have a different agronomist there, I guess, and don't have me. Here's what it looks like. Here's Illinois. Here's Nebraska. Okay. So final thing, genetic by agronomic technology. There's a synergism here. Modern era cultivars will have greater yield potential and yield than prior era cultivars on the basis of that. So I don't think there's any argument. The data shows that here. They also have resulted in on-farm yields having greater productivity, but that's come from the agronomic practices to allow greater expression of the genetic yield potential. And the yield difference between modern and old cultivars is greater when both are grown in the modern. Now, when I first published some stuff about this back in the 80s, I got some pushback from people saying, you're not being fair to the old cultivars when there's an interaction. I said, why should I be fair to something I'm never going to grow again? Okay. So it is G by A interaction, but it reflects the synergism between efforts that have improved genetic yield potential and agronomic potential. And you can see that here. Newest cultivar releases, as you raise high yield production environments, you get more expression. So what are we going to do in the future? How's my time? Okay. Well, certainly DNA sequencing, and by the way, that shows that, by the way, before I finish that, it shows that agronomic improvements have accounted for also a significant share. And by my estimation, it's 2/3 genetics and breeding and 1/3 agronomic and physiology improvement. I'll come back to that just at the end. So these are some of the things. First thing I started when we sequenced the Williams 82, that was a greatest thing since sliced bread, because now we can operate more effectively once we know where markers are and where annotated genes are. Just completed a 50,000 genetic SNP marker thing that was just released here this spring. The Soybean Board is funding 2 major projects that look at the identification of QTLs in the NAM project, nested association mapping, which they've done in corn. And ultimately, I've provided them with the DNA sequences of all those milestone cultivars. They're generating at Iowa this DNA sequences of all those major milestone cultivars to find out where breeders left their signatures in the soybean genome. Okay. I'll repeat that again. Breeders, when selecting for yield, had to leave some sort of signature in the genome, and we want to find out what in the hell that is and where it's at. So those are the 2 things right down there. Sunday. Just got the data to Aaron Lorenz, who's working with me on this project, and this was the first principal component analysis. You see the color blue is North America, China down here, and a couple of different spots in Japan and South Korea. So we're working on that to see what this is a lot of genetic diversity out there. Can we use any of it? You can see, if you compare these 2 charts, you can see the U.S. is just this much plus a few out there. So it's been sampled, but obviously there's a lot more out there to look at. Oops. And my interest, of course, has been in seed yield and protein. So I have seed yield on this axis, seed protein and oil on this axis, and percentage protein runs at a 0% moisture basis will be in this zone. So, and oil will be down here on the scale. So for each accession of these in the collection, you'll have 2 points here, one for its oil and one for its protein. It's kind of a hard chart sometimes I try to get across to students. So if you look out here, whatever this individual accession that's very high yielding, this was its protein and this was its oil. It's a double scatter plot, so to speak. Okay? And that's the line. So you can see here, even in the collection, as we go to higher yields, oil goes up and protein goes down. But are there any outliers that we can use in which we can choose that have the same yield but higher protein perhaps? and less impact on oil. So this is something Aaron and I are going to look at as well. The NAM project, basically we took 40 parents that were selected based on their diversity and mated them to this high-yielding public cultivar Iowa 3023. We have 140 lines in each of those 40 matings, so that's total 5,600. All those populations, plus checks, about 6,400 entries, were evaluated in single replicate trials at those 4 states in 2 years, plus there were others that participated only for partial replicate. And Brian calculates that the power to detect QTLs with 95% confidence, or that effect, I should say, can be as low as 3%. So this is we don't have one built for the NAM project. The NAM people used 25, I think, and they made the hub parent was B73. But this just gives you an idea. You self them all the way to F5, and then you do it. The pedigrees. So knowing what the pedigrees are, we can trace back where in the heck did those breeder signatures come from. Eventually, when we get the sequences all these. So we go back there and we selected these different cultivars. You can see their yield goes up. And by the way, this is a 2-year multiple location trial for these yields. So we damn well can predict with pretty good confidence that this is going to yield this yield this yield this, and so on. So if we sequence those, we should know what breeder signatures were implemented to change their genome. Likewise for maturity group 3 and 4. And when I read this paper back, or I didn't read it, I don't read human genetics, but I came across it in a report back in 2004 where they were looking for genetic signatures, breeder signatures, and they were using the human million marker plan to take a look at different areas of the population, those in Europe that adopted dairy farming versus those that did not. And so they're looking for the lactase gene, which is you're tolerant to lactase or you're not. And I saw that and saw this chart. And I thought, why can't we do this in soybean? And we now are, as I end my career, unfortunately. So what about the future? This is what Yogi Berra. Everybody know who Yogi Berra was? He was a baseball player, a catcher, then a manager, who had a lot of quotes that doubled back on themselves, like this one. I never predict the future, and I predict I never will. I love that. So we're going to look at the distant future. And you can see the predictions. Here's the data that we currently have that I operated on. And this would be the linear to infinity. This would be the logistic to its maximum as determined by nonlinear application of logistic regression. That might not be the one. We'll have to see how much it's developed. And this is the exponential equation if you fit an exponential to that. And those would be the years, these pink lines are years in which we finally reach 60 bushel for the whole USA. With the linear method, it's going to be around 2060. With a logistic, maybe around 2050, and of course with the exponential within 2035 or so. And of course there's been some predictions made. This is one with some rosy colored glasses, I think, that says technology is going to get us to this point. And if I look at this point, I'll be around when that happens, so I can make a bet. This one I won't be, of course. And these guys know nothing about agriculture, but just do modeling, economic modeling. Okay. And so they're predicting this down here. Now, if you're a betting man, I'm not one, but I try to know as much as I can before I make a bet. And a bar bet by definition is 2 guys at a bar, one of them who's knowledgeable and the other who's a fool. That's not saying these guys are. Just say they have less knowledge. And you have a 99% certainty of winning that bet. That's what a bar bet is all about. Okay. So I'm willing to offer a $10,000 check to any of these people or any people out here that by the time we reach 60, we're going to be here or here rather than here or here. And these guys, I don't consider worthy of betting because they don't know what they're talking about, even though they published it in PNAS. So, anybody out there wants to take it? For these out here, if I'm gone, I'll put it in some account, and you can either cash it if you win, or you can deposit it if I lose. Okay. All right. What's the impact? How do you rate the performance of two disciplines, breeders and agronomists? And I did soybean, and I'm going to come back to corn because soybean is the Cinderella and corn is the wicked stepsister, okay? that's been my modus operandi. Okay, so here's corn. Obviously, they've traced corn back to 1960. Come along here and bingo. Geneticists discovered that you could generate both heterosis and general combining ability, and ever since then it's been on upward, okay, as shown here. That's overall U.S. If you look at, oops, too fast. If you look at Nebraska, you can see that trend started here for irrigated and here for dry land. So, as I said, dry land is going to limit your genetic potential. There's no way of getting around that, whereas you can fully express it under irrigated conditions in Nebraska. So, you compare this, oops, I'm sorry, my finger's getting, with this what we have for irrigated and soybeans. And this I use in a drought, so I'm not going to talk about that. So you can see the irrigated is quite high. There's also a break point here, and I don't have the year on here, about 1985 apparently, where it's taken off faster. Oops, sorry. Now it wants to go fast. That it wasn't moving before at all. There. Can you move that for me? Oh, maybe I'm back here. Okay, here it is. So if you look at those two slides, and you can see this is corn yield improvement and soybean yield improvement. So soybean breeders out there, raise your hands. Just one? Where are all my graduate students? Or George's, like you said. Here's the soybean one, here's the corn one. Okay. You look at that and you can have, as I did one time, National Academy scientists say, soybean breeders and agronomists are doing really what he said, and mainly I interpreted a piss poor job. Really? That's coming from a basic research brilliant scientist who's applied research stupid. Okay? So let's take a look at this in detail. Okay? So we know that soybeans yield 3 to 3.5 times higher than corn. And so if you know that and you plot them on the same scales, I'll talk about why you should do that. You can see that the line is linear. So now the soybean breeders out there can pat themselves on the back without breaking the arm. Okay. Because over 40 years, this ratio, 3.25 to 1, is applied under irrigation in Nebraska. 40 years. Obviously, it's been some ups and downs, but the whole trend line, which is what I use to eliminate the outlying years, look at the trend line. Now, why is that? And so here's where I say, if you look up here, you can see 3.25. And also on a bushel per acre scale over here, 3.25. Well, actually, this should be 3.0. I made a mistake there. But based on this kind of evidence, I said down here that on-farm, irrigated Nebraska farm improvement, that's where you're looking at the genetic contribution in the presence of the best possible environment. When we do that, we can see that the genetic contribution, when I grew these tests in Nebraska under irrigation, I only had this rate of improvement, whereas the on-farm yield rate up here was 0.645. So this is the number I came up with. So breeders, soybean breeders can say, I definitely contributed 2/3 of every bushel per acre out there you have, producer. Okay. Whereas agronomists can say, I accounted for 1/3. At least based on my saying. And you can see that ratio has been stable over the years as well. So this is just a plot of that corn-soybean ratio. But you already saw that. This is for soybean. And so if we stop right here, we can see that why is this occurring? Why? This is what we put in a recent publication in a book that's going to come out from CSA. Why is corn and soybean, when you plot them on these 3-to-1 scales metrically, why are they equal? Well, corn has a jet engine, soybean has a prop. When it comes to photosynthesis, right? So, and we know that C4 makes for the best photosynthesis. And in fact, what we'd like to do is convert soybeans if we ever can from a C3 to a C4. Now, Tom's working on trying to improve its Calvin cycle enzymes, which will help out, and maybe even make us beat more corn, right, Tom? But corn is also mostly a carbohydrate. Soybean seed has this high protein and lipid that requires more photosynthetic energy to produce, and of course the nitrogen needed to reduce the nitrogen to protein. And both of them will use that carbon for lipid. Soybeans can also fix their own nitrogen, but it ain't free. Rhizobia costs the plant money, photosynthetic money. And not only that, for corn you have to provide nitrogen to corn. You don't have to do that to soybean. From what we discovered, they can, as long as you plant it, corn-soybean rotation, soybean can get all this extra nitrate it needs from what's left over. So here's the bottom line. C4 better than C3. C4 more, but C4 requires 2 more photons for every carbon you fix. It's got to be grown in when light intensities are high. That's why that evolved in the tropical savannas, because of the greater light period. C3 is less due to photorespiration. C4 system, you put your rubisco in the bundle sheath cells where it's not exposed. So this would be typical percentages to 84% carbo, 10% pro, and 5% lipid for corn, and these would be those. So when I used to teach physiology, I told students, when you grow corn, you just get a bag of flour at the end of the year. When you grow soybeans, you get a smorgasbord. But they would say, yeah, Dr. Specht, but with corn you can make ethanol or beer. So I lost that argument right there. But corn always yields more, okay? So I'll finish up with this. Some notable quotes from Yogi Berra. One of them was, the future just isn't what it used to be. I love that one because it's actually true for me now. I'm almost 70 years old. And by the way, the reason I'm wearing a coat and tie is because my brother once told me, you know, if you're choosing only n times to wear such a thing, there'll be an n1 plus time where somebody else will dress you. And you don't want people saying, my, I never saw him in a coat and tie, and he either looks bad or worse because of that. So that's why the future. Of course, you heard this one. I love this one because that's why I decided to work with Aaron, because I was lost, even though I was making good time. I need some bright young people out here to carry that forward. And I also love this one, because somebody mentioned about Tom Sinclair and Ken Katzman out there last night. In theory, there's no difference between theory and practice, but in practice, there is. So always be aware of theory and not take an empirical look at practice, because you'll get way off if you don't. Okay, with that, I'll conclude. Thank you all. Hopefully I'm right on time.
Yeah, with that, we'll have time for a few questions. So I can walk around the room. If you want to raise your hand, you can speak into the microphone.
And by the way, I love to be challenged, so if you think I'm full of crap, stand up. Part of the scientific arguments that make things better.
Thanks, Jim, for your presentation. Question, what do you estimate or calculate from all the various things that you've done might be the absolute maximum yield you might get if corn captured 100% of the light energy and soybeans the same? What do you think that maximum potential is?
360.
For yield?
That's my estimate, based on my interpretation. That's bushels per acre.
On beans or corn?
With a near perfect environment. I say, when you say maximum, that's what I call probably the practical limit you could get if you had a cherry-picked environment and intercepted all the light, and you didn't change, like lengthening the growing season on me or other things.
Yeah. You got a
For
ton
soybean, it's 120.
You have a ton of assumptions to make, but the national corn growers this year or last year, a gentleman had over 400 bushel yield, supposedly measured. Yeah.
Well, my response to that is, I'm a scientist. If you want to get those yields, see the magicians that did that. Because it doesn't compute. And in fact, Larry Purcell has checked out the producer in Arkansas, and he doesn't have the dry matter to get those yields without having to assume what do I call it, a 70% harvest index. Not likely. You checked out the dry matter on seasonal basis, and they'll be coming out of crop science.
Thanks for the presentation. My question is, why you compare this yield potential of soybean with maize? As we know that maize is a C4 plant and it will automatically have a great yield potential. Why not you compare the C3 plant with a C3 plant like soybean with a rice and then check the yield?
My hearing is bad. Can you interpret the question for me? I couldn't hear it up here.
My question is, when you compare the yield of soybean with maize, maize is C4 plant. Why do not you compare a maize with another C3 plant and check the yield difference? Yeah. My question is why you compare the C3 plant with C4 plant? Why not C3 plant with C3 plant? Thank you.
Sorry, I had to step away to the static a little here. So,
my question is, could you talk about what do you think about the hybrid soybean in the future, the effect about the yield improvement? Hybrid soybean. Yeah, I mean, what do you think about the hybrid soybean?
okay,
Thank you.
good question, actually. Well, there are 4 considerations. First of all, you have to document heterosis. That's number 1. And it has to be sufficient that the proprietary company can charge enough for it and the farmer will accept it in terms of how much yield they would get over a self-line. So that's an important aspect. It's going to be hard to do the pollen transfer in soybean because it has to be done by insects, and when it rains in July, as I discovered with my male sterols, you don't get pollination in soybean. And you think about the yield levels, you can plant a half bushel of corn per acre commercially or for producer fields, which means the number of fields you have to have in seed production versus the number of fields that are going to be in farm production, that ratio is a lot higher. In other words, how many seed production, hybrid seed production acres do you have to have versus the number you're trying to sell to? So if you get your inbreds, female inbred, when it's pollinated, produce 150 or more bushels per acre, that's a lot different than if your female is a soybean and you're only producing 50 bushels an acre. And you got 80 million acres to supply in both cases. That means your seed production fields in soybean are going to take up a lot more space to get the seed to the producers that want it if it works. So those are it's been patented for a while by somebody. It was a 2-restorer system, but nobody's ever used it. Cytoplasmic male sterile.
We have one last question.
Thank you, Jim, for the presentation. I don't know if you're familiar with the work in wheat on climate change, but one of the important predictions is that there will be decreased nitrate assimilation and we'll continue to see depressed protein concentration in the crop. Is there similar evidence in soybean that we'll see reduced protein levels as a result of increased CO2 concentration?
Well, that may be true for a cereal, but for a soybean that does nitrogen fixation, you have to remember for what nitrate it doesn't get, it trades off with nitrogen fixation. So if there is an effect like that, there would be a compensatory trade-off, I think. I don't know enough to do the experiments, but I would think that would apply. But so what you talk about would certainly apply for corn and cereals and other that can't do biological nitrogen fixation. But the biggest concern for me is that Nebraska may not be a soybean producing state in the future, they'll all be in Canada because of the global warming trend. As you can see, North Dakota went from just a few acres to a couple million acres in just a few years, and Manitoba went from 0 in 1985 to, I think, 500,000 acres. I don't have the numbers with me, but I didn't show the Canadian charts. They're big-time soybean producers in Canada now, so if you look at the last record, so soybeans are going to move. If you can't grow them here, they'll grow them where they can. That's what I call adapting to climate change. That's happening even though the IPCC wants us to mitigate climate change. We're doing an awful lot right now to adapt to it.
All right, let's thank Dr. Specht for his time. Okay, right now